Hello there. We're going to give it a minute or two for people to continue to join the webinar. So just sit tight and we'll get started in just another minute. All right, the participant numbers have stabilized a bit, so I think we can get going here. Thank you for joining us. I'm Rex Malmstrom. I'm a scientist at the JGI, and I'll be hosting today's session. And we're going to highlight some of the capabilities of EMSL and JGI and explain how they can be accessed through a single proposal in order to advance your own research. It's our hope that this presentation will provide an opportunity to get some answers to your questions, whether they're specific to the technologies of either of the facilities, or if they're more general regarding the process of applying to become a user. We're going to collect the questions through the Q&A feature of Zoom, and we'll take some time at the end of the presentation to address them. And so just anytime you have a question, please just enter it right into the Q&A. And then we'll capture your questions and make the answers available after the fact for those people who can't participate live, and you'll be able to rewatch this video. Over here, this is a quick outline. I'm just going to do this brief introduction, and then we'll move on. Okay, so before that though, just a little bit about us. The Joint Genome Institute is a U.S. Department of Energy user facility, and we're located at the Lawrence Berkeley National Lab. We're world leaders in the genomics of plant, fungi, algae, and complex environmental microbes. And we help researchers characterize how these organisms and communities carry out their complex biogeochemical processes, and also how they can be harnessed for sustainable biofuels and bioproducts. The JGI has resources and expertise in the areas of DNA and RNA sequencing, DNA synthesis, metabolomics, and data analysis capabilities. The Environmental Molecular Sciences Lab, or EMSL, is also another DOE user facility, and they're located in the Pacific Northwest National Lab. EMSL has expertise in a variety of areas too, including imaging, proteomics, chemical analyses, biogeochemistry, and modeling capabilities, and to just name a few. We're going to hear more about some of these capabilities in today from EMSL's Mary Lipton and JGI's Tanya Woike. And we'll also have a talk from Michelle O'Malley, highlighting how she has collaborated with us to advance her own research program at UC Santa Barbara. And then after Michelle's talk, there'll be a chance to address your questions. So scientists like you can access either user facility through different calls for proposals throughout the year, and the proposals go through a peer review process. And if they're successful, they let you draw on the expertise and technologies of JGI and EMSL. So this is important. I'm just going to repeat that in contrast to other agencies where accepted proposals are awarded funding, when it comes to EMSL and JGI, successful proposals are awarded free access to resources at the facilities. A few years back, EMSL and JGI have created a FICUS program that was meant to allow researchers to apply to both user facilities with just one proposal. And this program has expanded over the years to include other resources such as BioSANS, the Biological Small Angle Neutron Scattering Instrument at Oak Ridge National Lab, as well as include some resources from the NSF-funded NEON program. We'll hear more about BioSANS and NEON in separate webinars in the coming weeks. I'd like to point out the letters of intent for the FICUS program are due on March 17th. And so that's approaching quickly. However, this is just the letter of intent stage, and it's possible to modify proposed work between the LOI and the full proposal submission. So make sure to get your foot in the door by submitting the LOI. Okay, I'm going to turn it over now to Tanya Woike. She's the JGI's Director of User Programs, and she'll provide an overview of some of the technologies that you can utilize when working with the JGI. The last thing I'd like to say, though, is that I encourage you again to submit questions via the Q&A tool as we go, and we'll do our best to address them in real time. Okay, Tanya, the stage is yours.
All right, let me share my screens. And all right, can you all see my screen? All right. So first, a quick introduction. My name is Tanya Woike. I'm currently filling in as the interim deputy director for the user programs at the JGI. And as Rex mentioned, I would like to spend the next 5 to 10 minutes on giving you a brief overview on the resources that we offer at the JGI for the FICUS program. Now, before talking about the actual resources and capabilities, let's have a look at how part of JGI is structured. We have our Genomic Technologies group, which sits at the core of the JGI. This is a group that does anything from developing new molecular biology workflows to improving processes, making them higher quality, more cost effective, and to running our many production projects from sample receipt through Sequence QC and beyond. In addition to this group, we have the science programs. These are the plant program, the fungal algal program, microbial program, and the metagenome program, as well as the DNA synthesis science and metabolomics programs. And where I'd like to envision that is that the science programs are sort of a connector between the JGI Technologies group and the respective user communities. The science programs also facilitate much of the downstream data analysis, for example, through the comparative analysis systems that they develop at JGI and maintain. The important takeaway message sort of from the slide that I would like you to walk away with is that the integration of genomic technologies and the science programs aligned with the needs of our user community and the DOE mission shape the products that we do offer at the JGI. And these products are sequencing, DNA and RNA sequencing, DNA synthesis, and metabolomics. And proposers can request these for DOE relevant projects that involve plants, algae, fungi, microbial communities, bacteria and archaea, and viruses. Now, the size of the request that you can request through a FICUS call has some limitations, and those are for sequencing 1 terabase, for DNA synthesis about 500 KB, and for metabolomics up to 200 samples for polar and 500 samples for non-polar metabolomic analyses. And what's important to mention here is that successful proposals often exploit a combination of different capabilities in addition to the required integration with the resources that are offered at EMSL. Now, let's briefly talk about these resources in more detail, and I'm going to stop my video because my computer is running hot. Our core sequencing capability, our core capabilities at the JGI are de novo genome sequencing and adaptation of plants, algae, fungi, bacteria, archaea, and viruses, as well as transcriptome sequencing. For microbial communities, we likewise offer DNA and RNA sequencing through metagenomics and metatranscriptomics. And also part of our core capabilities are resequencing for variant detection. And this could, for example, be a laboratory evolved microbial populations. Our workhorses are the PacBio Sequel II and the Illumina NovaSeq. And we used to have rooms full of sequencing machines. Nowadays, it only takes a few. So we have 3 PacBio Sequel II, and we have 1 NovaSeq instrument. And you can see those here on the right-hand side. And in the bottom picture, you can see a NovaSeq flow cell. As flow cell produces about 3.4 terabases. And if you were to run a NovaSeq 24/7, you would generate more than 500 terabases per year, just to put this in perspective and letting you know what these machines really put out. We also provide, skip to the next slide here, more custom capabilities. And those include, for example, fluorescent activated cell sorting, not only for traditional single cell genomics, but also for targeted single cell genomics and targeted metagenomics or mini metagenomics. And on the bottom left-hand side, for example, you can see a picture of our flow cytometers. Custom capabilities also include stable isotope-enabled metagenomics, DNA methylation analysis, and chromatin analysis. through ATAC-seq and ChIP-seq. For ChIP-seq, the DNA needs to be immunoprecipitated by the user. And if you're interested in methylation analysis specifically, I would encourage you to join our upcoming webinar on long-read metagenomics and methylation, and that will be on March 10, so please check it out. As for our DNA synthesis capabilities that we offer gene and pathway DNA synthesis, whole genome guide RNA library construction and QC, as well as organism engineering. And here as well, we had last summer a webinar on July 8. So if you're interested in our synthetic biology capabilities, please check out that webinar. It's available on the JGI website, and it educates you very well about the capabilities that we have and how users use these and exploit these for their own research. Lastly, our metabolomics capabilities. What we offer is LC-MS-MS-based metabolomic analysis of both polar and non-polar metabolites for functional genomic studies. And we also use these data and we help you integrate them with the genomic data. So we have platforms and tools to do that. Again, if you're more interested in metabolomics capabilities, please check out our engagement webinar that we had on November 5 last year in 2020. There's a great overview from Trent Northern and Ben Bowen on the JGI metabolomic program and the capabilities we offer. And Jenny Mortimer, a scientist who used these capabilities, gave a nice overview on how she integrates them into her own research. And with that, I would like to encourage you to put questions in the chat. Please also follow us on Twitter and on LinkedIn. And up next will be Mary Lipton, and she will be giving you an overview on the available resources for the FICUS project.
Thank you, Tanya, for the introduction. Let me share my screen. And my name is Mary Lipton. I am an integrated research platform lead in the Environmental Molecular Sciences Laboratory. And today I'm going to talk to you about some of the resources available in EMSL for the FICUS projects. EMSL is organized into two different science areas, the biological science area and the environmental science area. And each of the science areas are organized into what we call integrated research platforms, representing a different aspect of available experimentation and resources in EMSL. What I'd like to do today is provide a brief view of each integrated research platform and how they can be used in FICUS research. The first integrated research platform is the Biomolecular Pathways platform, which I lead, which aims at discovering the underlying principles that translates genomic information into metabolic pathways and regulatory networks. It's one of our hallmark platforms in EMSL that many FICUS users take advantage of, and it consists of a number of different types of analytics. What we're focused on is the latter part of sort of what Tanya talked about, is looking at integrated omics, from proteomics, lipidomics, metabolomics, where we can take one sample and get all of the of these biomolecules out in a single extraction. So we can get lipids, metabolites, and proteins and apply it to a wide variety of different instrumentation. So we have a comprehensive suite of metabolomic measurements, including NMR, GCMS, liquid chromatography mass spectrometry, ion mobility mass spectrometry, and other methods for doing metabolomics. We also have bottom-up proteomics as well as intact protein measurements that allow us to look at post-translational modifications as well as protein complexes. Our next integrated research platform is led by Scott Lee, and it's the structural biology platform that is focused on generating structural, biochemical, and dynamic information about biological materials, protein, and protein complexes at the nanoscale, spatial, and temporal resolution. Using a combination of high-resolution electron microscopy and NMR, we can determine the structure of soft biological materials, proteins, and protein complexes. We also combine the structural biology instrumentation with high performance computing to expand these analyses to do correlative multimodal imaging and analyses. The next integrated research platform is a cell signaling and communication platform. And what we're looking at, what we're aiming to do with this is understand how complex phenotypes arise from individual molecules and their interaction with other molecules, and look at communication between cells. One of the newer capabilities we have in the cell signaling and communication platform is the ability to combine laser capture microdissection extraction of single cells or collections of cells with our nanopots sample processing. Nanopots processing allows for a very small amount of material to be extracted and then processed for either omics characterization or imaging, say using FISH. And so we're actually able to get down to the single cell resolution level and look at what's happening at single cells or single eukaryotic cells or collections of prokaryotic cells. One of our newer integrated research platforms led by Amira Kami is our plant and ecosystem phenotyping platform. And what we want to do here is dissect the genotype to environmental interactions using plant microbe crosstalk in the rhizosphere region, as well as look at plant and ecosystem phenotyping. What we have available is a combination of plant growth chambers equipped with in situ phenotyping measurements that allow you to look at the phenotype and within the growth chamber without having to remove the plants and disturbing the plant growth. We're also able to do rhizoboxes and rhizotrons so we can look at plant growth and the developing roots without actually disturbing the plant. And then we can open up the rhizoboxes and actually sample what's going on in the soil along the roots for subsequent analyses, again, with a minimal disturbance of the roots. A lot of these plant growth chambers are equipped with the ability to pipe in a stable isotope labeled gas, or we can use stable isotope labeled metabolites in the growth media or in the added to the soils to track metabolite and substrate flow, along with and in combination with some of our other analytics and our other integrated research platforms. So our last two integrated research platforms is our biogeochemical transformations and our ecosystem interfaces. And the biogeochemical transformations platform led by Mark Bowden is looking and examine the detailed molecular biogeochemistry in microdomains in soils and sediments, whereas the ecosystem interfaces is looking at tracking the flow of nutrients and materials that within these biogeochemical processes at the ecosystem interfaces. One of our most highly subscribed analytics in biogeochemical transformations is using mass spectrometry and NMR to characterize the organic matter analysis in soils and sediments. And this is a capability used by many of the ficus projects that we already have. But we also have some newer capabilities to be able to say use solid state NMR based analytics to look at the structure of fungal and cell walls while it's still structured, rather than having to deconstruct the cell wall and infer what the structure looked like. And going back to what we can do in the phytotrons is add the isotopes. We then can use a lot of the isotope analytics we have under the ecosystem interfaces platform to trace the stable isotopes through, say, a plant into the roots and into the soils to find out what those flow of materials looked like and what they actually, where they actually went in the cells, what kind of biomolecules were the tracers then turned into through our metabolomics, and where do they end up either in the soils, in the microbes, in the roots, through some of our isotope ratio mass spectrometry capabilities. And so like Tanya will suggest that you put the questions into the chat, and I would like to introduce Michelle O'Malley from UC Santa Barbara, who will talk about her ficus user project of unlocking the biotech potential of anaerobic fungi through ficus. And I thank you for your attention.
Great. Thank you, Mary. Let me just share my slides here. Okay. Can everybody see the slides? Okay. All right. So it's my great pleasure to join you virtually today to tell you about actually 2 ficus projects that my lab has been associated with. And both of these are going to focus on unlocking the biotech potential of a weird group of organisms called the anaerobic fungi. And so just to set the stage, to give some scientific background why we're interested in these unusual fungi, we got very interested in looking at anaerobic microbes. So these are microbes that thrive in the absence of oxygen. Chiefly, we were interested in anaerobic microbiomes where there's a wealth of organisms that are secreting powerful biomass degrading enzymes that turn ingested lignocellulose, say in a large herbivore, to short chain fatty acids and ultimately sugars. And there's a lot of things happening in these communities, of course, but we and many other groups around the world have been really excited to attack these problems and understand what's going on in these weird microbiomes because of the new development of omics tools that now enable us to really get inside these communities and figure out what the biological processes are and the systems biology that underpins these lignocellulosic conversions. And my lab in particular is interested in harnessing the power of these anaerobic microbiomes for biotech and for biomanufacturing, for taking waste material and turning that into something value added. And so what I'll tell you about today are 2 ficus projects that my lab was involved in. The first was actually one of the first ficus projects in 2013 about identifying the enzymes that come from anaerobic fungi. And the second is much more recent and still active, which is about deciphering the structure and function of some of the natural products and secondary metabolites that are made by these anaerobic fungi and how they're impacting microbial communities. I'll take just a second and tell you a bit about the anaerobic fungi. Suffice to say, I could sum up this whole slide by saying that they are very weird microorganisms. They're early branching fungi. There are 9 or more characterized genera of them. But what's important to note is that they reproduce from a spore stage, and then that spore goes and germinates into a root-like structure, and that is where we suspect that a lot of the biomass degrading enzymes are being degraded and are being secreted to degrade lignocellulose that these fungi are burrowing into. And that process continues until more spores are created and released. What's important to understand is that due to this unusual life cycle and structure of anaerobic fungi, it makes working with them very tough on several levels. In particular, there's a lack of genomes and available strains to work with them. So we and others who work with anaerobic fungi generally go and isolate our own strains from herbivores. There's a whole bunch of complexity with growing them that I won't get into. They're difficult to store. They are not yet very genetically tractable, although this is changing, and no proteomic characterization when we started working with these fungi, and a very small community of researchers. So this is really a great jumping off point to leverage a lot of the strengths of JGI and EMSL to try and attack some of the scientific problems that we wanted to tackle. And this is just a helium ion microscopy image. This is actually colored for a bit of an effect, where you can see, this is taken at EMSL, where you can see these fungi working up close and burrowing in with lignocellulosic biomass. I won't go through how we get these fungi, but suffice to say, we go out and visit the animals, and they're donating some fecal materials, of which we apply pretty old school microbial enrichment techniques to gather isolates of anaerobic fungi from these sources. These are 4 different isolates that actually the JGI has since genome sequenced. I'll get a little bit into how we did that with them. And these are helium ion microscopy images taken at EMSL that shows you each of these 4 different strains and how they are literally punching into plant biomass. Okay, and so the first story of this of the first ficus that we were involved in led to the publication of this paper in Science several years ago, which is about essentially counting the broad enzyme systems in these anaerobic fungi. And what got us excited is that we were able to work with JGI through this first ficus project to not only sequence the genomes, which was tough, but to initially just sequence transcriptomes from isolated RNA, and then also doing RNA sequencing and differential expression analyses to understand how these fungal isolates were responding to their environment. In short, we were really interested in identifying novel enzymes, and we knew that there was probably some response to these fungi as they're growing on different substrates. So when they're growing on glucose, there's no need to make biomass degrading enzymes, but as you take sugars away and start challenging them with cellulose and lignocellulose, they start to fire more enzymes. And so we worked with JGI through the transcriptomic pipeline that was mentioned to actually develop a pipeline for identifying novel enzymes. So we did this by growing fungi with reed canary grass to fire all of their lignocellulose degrading enzyme machinery. And then after some time, we pulsed with sugar to repress expression. We were able to do a differential expression analysis. I won't get into all of what this means, but essentially all of the lines here you see are transcripts. Everything that is red goes up in time. Everything that is blue goes down. And we were really interested in the transcripts that get repressed when sugar is introduced. And this allowed us to really pinpoint a lot of enzymes and enzymes that we didn't know about that were associated with biomass breakdown. And we were able to find novel biomass degrading enzymes here by combining the strengths of JGI's transcriptomic pipeline with proteomic verification at EMSL. And in fact, we were able to discover a novel set of fungal scaffolding enzymes that are critical to the assembly and the function of fungal cellulosomes. And we talk more about that in this Nature Microbiology paper if you're curious on details. Since then, we worked with JGI to actually sequence the genomes of anaerobic fungi. The first anaerobic fungal genome was sequenced by Mustafa El-Shahid's group in a publication that came out a few years ago. And they were able to, and that was a gargantuan feat in and of itself, but you see a measure of genome quality is generally the number of scaffold fragments or the pieces of the genome, which were quite high. Since then, through developments in the field and the expertise and the instrumentation at JGI, we've been able to work with them to sequence 4 additional genomes where we're really pushing the number of scaffold fragments way down. And I understand that there's a couple more genomes from other groups in the pipeline that really improve the genome quality for this group of fungi. And this is important because it allows us to really leverage the genomes for lots of different things, from biotech to comparative genomics and just understanding the basic biology of these fungi to develop genetic engineering tools as well. Leading to the second project, I'll touch on briefly, which is still active, this ficus project. Sequencing the genomes allowed us to see what else is in them. And one of my students, Candace Swift, who's recently just graduated, was scanning the genomes and realizing that there's actually a lot of genetic building blocks for secondary metabolites. So these are usually associated with everything from differentiation to cell maintenance to cell competition. A lot of antibiotics, for example, are secondary metabolites. And so just by scanning through each of these 4 sequenced genomes at the JGI with a tool called Anti-SMASH, we were able to find many different what are called gene clusters across these 4 fungi that are actually transcriptionally active and might lead to the production of hundreds of likely natural products that could be useful for biotech. Working with JGI Metabolomics and specifically Trent Northern's group, we were able to take a lot of these native fungal cultures and fractionate them to get at what the actual metabolites are. So I won't get into the technical details here, but we found different peaks of interest by comparing cultivated fungi against controls. This starred compound here is something that we found quite early on, which is a secondary metabolite called Bauman, which is has actually been previously identified in in other fungi, so-called higher fungi. It's an antioxidant, but this is the first report of any natural products, the secondary metabolites being found in the anaerobic fungi. So we were very excited about that. The other thing that we are actively doing in this project is to actually use gene synthesis capabilities at JGI to synthesize many of these gene clusters that have been identified in the anaerobic fungi for production in model hosts like yeast and the fungus Aspergillus nigelens. And we are using expertise and instruments at EMSL to verify the proteomic expression of many of these cloned gene clusters, and then also leveraging expertise at JGI and EMSL to really get at the metabolomics analyses of what's going on in these what's being made by these biosynthetic gene clusters. And again, I'll spare you the technical details, but to suffice to say that we want to use different instruments that will tell us if these gene clusters are actively being transcribed, and then and made into proteins, and if so, what small molecules are they making? And that takes lots of different analyses from mass spectrometry analyses to also NMR. And we like this type of approach in leveraging the two facilities because it really opens up what we're able to do and the science we're able to do as part of this project. And in particular, it helps us by expressing in two different hosts, which was enabled really by gene synthesis at JGI. We can really expand the search space for these novel natural products. And through complementary strategies and having just different expertise and different instruments at both JGI and EMSL, we're really mitigating project risk. I'll explain that by saying that those who work on natural products know that many of these heterologous expression efforts fail. And so we're just trying to develop a path forward where we're learning about the basic biology. And even if say one clone fails and it doesn't express well, we have a whole set of others to rely on its screen. And so I'll wrap up by saying that I hope that the science that I've shown you has been insightful. But in terms of engaging with ficus, what I would recommend having two projects that we've been involved with is to talk with the scientific leads early and often. They're there to help you. They want you to develop a very strong proposal, and they fully expect to have conversations with users. And in my view, this is really the only way that you can be sure that you're matching your science to the capabilities that are offered through JGI and EMSL. And it's really the best way that you can find out state-of-the-art advances and ways that and instruments that you may not know what they do, but the scientific leads do, and they can help tailor your request to the appropriate science. And no one expects you to be an expert in every single technique that you might want to make use of. The experts are rather at both facilities and there to help you and to train you and your students and trainees if they are involved in the research. And I'd say also it's important to have DOE relevant science questions. This has been stressed a bit, but you want to make sure that the research that you're proposing to do and that it's enabled by these facilities is also relevant to DOE. Preliminary data is not necessary, but it really does help, I think, with proposals that are very competitive to show that you can generate high quality samples for analyses. This might be DNA or RNA, but it could also be protein samples for proteomics. Essentially, this is just the guarantee that the request that you make can actually be fulfilled. And watch for key dates. Remember to submit a letter of intent. I know that the dates have been shared already. And an added sort of personal note is I found it very rewarding to involve trainees in this process of writing the proposal and certainly in doing the science. I think that it helps to spread the word about these user facilities, and to get trainees involved in the process is great. And with that, I will thank all of the trainees in my lab who have submitted all of these samples. I'm not of very good use to my lab other than to provide advice, and I'm not physically generating these samples, but in particular, a lot of the work that I've told you about today has been done by Kevin Solomon, who's an alumni of the group, is actually at the University of Delaware running his own research group, as well as Charles Heitzema, another alum for the lab, and Candace Swift, who is seen here on the more recent ficus proposal. And of course, all of our funders for support. And I'm happy to take any questions and transition to discussion.
Thank you, Michelle. That was a fantastic presentation. I'd invite if you could stay on right now, that would be fantastic. I'd also invite Tanya and Mary to come back on if you can get your cameras or your microphone. So we'll see if we can do some of these questions. I've had a number of questions coming in so far, and I'll just start distributing some of these questions. So the first one actually is to Mary, related to some EMSL capabilities. For the Rhizoboxes, do users send samples to EMSL? Do they have to send the plants that are already grown, or maybe can they do seeds?
Either we can grow the seeds here, and we have the capabilities, we have the growth chambers and the capabilities to grow them. If you have the capability to send a Rhizobox, we can work with you to help you extract whatever we want, whatever you'd like to extract. So there's a number of ways of interacting with the EMSL on the Rhizoboxes, and we would look forward to working with everybody on figuring out what works best for the individual researcher.
Great. Can you also tell a little bit more about the single cell omics capabilities? There's a question of someone working with dinoflagellates and so single cell algae.
Yeah, so what we've done is we use laser capture microdissection, say on a plant where we want to isolate a single cell, but we can also do cell sorting to get a small number of cells or a single cell. And then it goes into our nanopots sample processing. And I honestly don't remember exactly what nanopot stands for, but what we've done is miniaturize the sample processing and eliminated a lot of the sample transfer, sample plastic. So it's all done in a single capillary, and we have very, very small sample loss. So we're actually able to process a single eukaryotic cell from lysis to biomolecule extraction to analytics, all within that capillary. If we're not going to do an omics characterization, we can take that capillary and apply the cell to an imaging platform to be able to get those images. But it's basically a miniaturization of our sample processing so that we can get down to the single cell limits.
Excellent, thanks. Tanya, do you want to add anything about single cell capabilities at JGI?
Yeah, so at the JGI, we have various single cell capabilities. The most traditional one is single cell genomics for bacteria and archaea. That's actually run in Rex's group, in the microscale application group. For that, we have our PIs send us cell material that's cryopreserved in glycine, betaine, or some cryoprotectant that gets shipped to the JGI, that then gets single cell sorted with FACS at the JGI in 384 well plates. The single cells get lysed, whole genome amplified with MDA, and then we can do sequencing on them. We don't do 16S identification, so they get sorted sort of blindly, whatever amplifies. That allows us to also sequence these so-called taxonomic blind spots. So that's one product that we have, and more sophisticated are these targeted approaches that I mentioned. One example is BONCAT, which allows us to kind of light up and sort translationally active cells. And we can do then either single cell genomics or more popular sort of mini metagenomics, where we sort hundreds or thousands of BONCAT labeled translationally active cells that can then be sequenced as this kind of enriched mini metagenome. So these are more custom capabilities, but these are some of the things that we offer at JGI.
Great, thank you, Tanya. I see that a few questions specific to Michelle O'Malley's talk have come through the Q&A, and she's been answering them there herself. I just saw a new one. So there was a question though about constant conversations that Michelle, that you had suggested, that have conversations with people at JG and EMSL. How can people do that? How do we know who's the person to contact? So this is a question for Tanya and Mary.
So I guess I'll start. The best place to start is with our website, and a good starting point is Nancy Hess and Scott Baker, who are our science area leads. But that doesn't mean that you can't go directly into one of our integrated research platform leads. We're all listed on the website, and we're also, I believe, listed in the call. And so please feel free to email any one of us with questions on how to access any of our capabilities. And if the person you contacted is not the correct person to answer the question, we're very good at passing emails around to make sure that we get the question answered by the person with the right expertise.
And that's very analog at the JGI. I'm listed as one of the point of contacts. Always feel free to get in touch with me. I will parse out these questions as needed to our programs. I showed you the structure of the JGI. We have these science programs. So they get parsed out to people that are knowledgeable in these areas, and that can help you.
The other thing I'd like to just point out is that I did have contact information on all the slides for each of the integrated research platform contacts. So if you go back through the recording, you can also find our email addresses there.
We'll also have a few emails on the slide at the very end of today's presentation. You can go back to the webinar and you can do things like Google JGI Tanya, JGI Rex, and so Marilyn. Get a hold of us. I'm just repeating. We will answer your questions or try and put you in touch with those people who can. Okay, so a question about eligibility. 3 different questions about this. Are postdocs eligible to be, can they submit proposals? Can international researchers submit proposals? And can we take proposals from startups?
So I don't believe through the EMSL process that postdocs are eligible. We usually like the proposals to go through a staff member so that we know postdocs are by definition transitory. So we want to make sure that the person's actually going to be at the institution that has started the proposal through the entire lifetime of the proposal.
International students?
Yeah. International startup. Okay, startups. As long as the data is not proprietary, as long as it can be published, anybody can access EMSL. The big thing is that the data has to be open. We can't do proprietary data that is going to go into a patent or be kept secret as part of our as part of our charter. And what was the third part of that question? Sorry, my memory's going.
Are people from international institutions
Yes.
eligible?
We work with we work with people all over the world. And so you do not have to be in the United States to access, I believe, either JGI or EMSL. And I'm answering for Tanya, sorry.
Yeah.
I wanted to, I just got some information that's coming from EMSL on the side that actually this year, new to this year, postdocs are eligible to be PI. So that's a new feature. And it's news to me too. So yeah, just to update that feature, postdocs are eligible.
And maybe one additional note from JGI for postdocs, we have, that is outside the ficus and it only applies to the JGI capabilities, but we have what's called the CSP New Investigator Call that's specifically targeted at new investigator, which also many of which are early career, which includes postdocs. They are encouraged to apply through that and use that as sort of a diving board to, build their own lab. So that if you're only looking for JGI capabilities, that's a good introductory call.
Great, thank you guys. I've got another question here. It says, apart from instrumentation support, how much help can users expect for data analysis of the different omic analyses? And are users expected to be trained to handle the huge amounts of data for each of these different analyses?
So that's a great question. We will take the project from sample submission through the data analysis, and a lot of times through the statistical analysis of the data. And so we run the data through the instrument. We will do whatever data processing is needed to take the ones and zeros from the instrument to turn it into biological information. And then we have statisticians on site who will actually do some of the normalization and introductory statistics. What we don't do typically is the biological interpretation, which is what I'm assuming that the investigator would do. But we will do a lot of the data analysis. We will also work with the user to train them on how to do the data analysis. So for example, we have a user who has who looks at circadian rhythms, and they want just raw process data to put through their own analytics, and they've developed their own analytics around our outputs. So it varies as to what the sophistication of the user is, but we will make sure that they get data that they are comfortable in interpreting.
And I would say for the JGI, once again, it's quite analog. It depends on the product. For our de novo genome assemblies, we do the QC, we do the assembly, we do annotation specifically for eukaryotes. There's a lot of curation in the annotation process. We add these data into our comparative genomic analysis platforms, for example, IMG for microbial genomes and metagenomes. And depending on the product, again, for other products like resequencing, we have pipelines where we map the data to detect SNPs and small indels, and we output that in a table and provide to the user. For RNA-seq, we take a reference genome, we do the mapping, and we provide that information to the user overlaid with the annotation. So there is a level of analysis, basic analysis that we provide. Now, again, data interpretation is up to the PI. More custom analyses are up to the PI, but we're always there to help. You can always ask us to help if we.
Just to chime in from just really quickly from a user perspective, I think it's a great opportunity to learn, right? Because this is why I mentioned having trainees involved in these projects is a great way for them to learn more skills. I mean, certainly as a PI, having a lot of people come through my lab who are doing sequencing projects and proteomics and metabolomics, it's really led to new marketable skills for them that they would not have if they were only working in my lab without these engagement opportunities. And I think it's just the scientists at both facilities are very open. And, you know, I think to call attention to the fact that there's also, if you want to become a really expert user in a lot of these techniques, both JGI and EMSL offer a lot of different workshops and training modules that are given throughout the year that, you know, if you want to go from really not having done, say, sequencing before to being a metagenome specialist, and there's ways to working with JGI and getting the training to really become that. And I think that, yeah, but that you don't need to be an expert in any of these things from the start. You just need to kind of pitch a project that's the right scope to get started.
Yeah, thanks for pointing that out, Michelle. We do have training opportunities that are under normal circumstances on site. And these are usually quite full. And they, as Michelle pointed out, the participants of these training workshops range anything from very entry-level students, undergrad, grad students, to more senior PIs. And there's really no pre-requirement of being an expert in annotation or metabolic reconstruction. So.
Great. And also to follow up on some of those webinars or some of that training that has been done online in this last year, and I think recordings are available, for example, how to use some of the IMG tools about microbial and microbial community genomes, annotations. And so that's also a resource you can look for too, is to look at these recordings of some of the trainings. Michelle, I have a question here that just, looks like you're typing your answer. So go ahead. We'll let you just go ahead and answer that question since you're doing it already. Okay. Let's see. I have another question here about the programs in general. Are there limits to what can be requested And do those limits depend on how different areas are coupled together, like metabolomics and single cell, but not gene synthesis, for example?
Yes, I can chime in on from the JGI side. So as I showed in my slide, there are limits to what you can request: 1 terabase of sequencing, 500 KB of synthesized DNA, 200, 500 metabolite analysis, depending on if it's polar or non-polar. Now you can mix and match. In fact, you're encouraged to mix and match, but don't exceed those ceilings. And I think the question also had a component of more early or entry level early career versus more senior PIs. If there's a delineation, we don't really delineate that within the proposal. But as I mentioned earlier, if you have a smaller scale, fairly smaller scale proposal, you may want to, and only are looking for JGI capabilities, you may want to look into the new investigator call. There, the scale is generally a little bit smaller. We have limits, for example, 72 samples for resequencing, 54 for RNA-seq are sort of the ceilings. So we don't expect you to put a full-blown multi-PI, fairly large scale proposal forward.
And if I can just chime in, our limits are a little more fuzzy. And typically what we like to do is have potential users work with our staff and work with our experimental scientists and our IRP leads to determine what an appropriate scope would be. Because when you start mixing and matching analytics and you start mixing and matching different approaches from the different IRPs, the ceilings become very, they vary a lot. And so if you come in just wanting to do, say, proteomics and metabolomics, it could be very different than if you want to do nanopots and do the imaging and the proteomics and say fish analyses. So because, as Tanya said, in EMSL, you can also mix and match in a variety of ways, the limits become a little grayer. So what we do is we encourage people to contact someone at EMSL or a number of people at EMSL that will help you appropriately scope a proposal that is going to be competitive.
All right, thank you for that. Let's see, I have just a few more questions and then it's time to wrap up. So let's see here. Oh, here's a quick question. So for the FICUS proposal specifically, do users have to request resources from both EMSL and JGI?
Yes, they do. And there should be, I mean, it doesn't have to be equivalent, but it should be a fairly substantial contribution from both user facilities. The FICUS were made to bring both user facilities together to bring to bear on each of the user proposals. And so what we don't want is someone to request all the sequencing they can possibly get and then run 3 proteomic samples at EMSL or vice versa. And so we really want to see that combination and the way that the user facilities can come together on a project.
Thank you, Mary. Tanya, a question for you. The letters of intent are due on March 17th. Could you say how those LOIs will be evaluated and then also when the full proposals will be due?
Yeah, so you will hear back by April 5. Full proposals will be due on May 3. Now, as far as how they are reviewed, first there will be a technical review. We want to make sure what's being proposed is technically feasible. The technical review happens in-house at JGI and EMSL. And then the scientific review is done by an external expert panel of reviewers, scientists, that assess the work for DOE mission. I have to say in the letter of intent also we're reviewing these for DOE mission relevance. And then scientific merit is very important and just overall feasibility of the work.
Great. Okay, last question here. How long do projects typically run, and do applicants need to have the samples already collected, or can they still, if they're doing field work, can they propose to do work on samples they're going to say collect in the next year?
A typical project runs for 2 years, at least at EMSL. And a lot of times the samples, the investigators have the samples ready already. There are some times where they do not. If you're doing if the projects will start October 1st, If your field site isn't going to be ready until April 1st or sometime in the spring, then that's when the samples will be ready. So we will usually, JGI and EMSL will usually have a kickoff meeting sometime in the fall to figure out what timing looks like, what samples look like, what the and what the work scope looks like, but it doesn't necessarily have to start on October 1st that you're sending samples.
And then to add on to that, from JGI side, the clock ticks a little bit longer. We give users usually 3 years, but for the FICUS, we want to be well aligned with EMSL.
Well, great. Thank you very much. That's all the time we have questions for. If you have any more questions, please feel free to contact any of the panelists. Send us a message. That will be, and we'll do our best to get to it. All right, not our best. We will answer those questions. I just want to pop this slide up again, just to remind you the letters of intent are due on March 17th. Here are 2 web links that you could use to find out more about the FICUS proposal in general, and you can learn about a bunch of the other capabilities, other calls that we have throughout the year. So before we end, though, I just wanted to say thank you to our guest speaker, Michelle O'Malley, as well as Mary Lipton and Tanya Woike, and to the other panelists behind the scenes trying to help with this webinar today. I'm sorry if we didn't address all the questions, or if we didn't address them completely, just let us know. We will do our best. We will not do our best again. We'll answer all those questions, and we plan to post this video as well as the questions and the answer that we gave on our websites shortly. So that's another place to go back and look. You can, again, if you want to learn more about the proposals, check these websites out here. And you can you can contact us with some of these emails, and you can stay in touch. You can follow us through the different social media avenues. You can see videos. JGI runs a couple different webinars focused on the science we do, and those are, I think they're pretty interesting. So you should think about checking them out if you have a chance. Okay, well, that's it for today. I hope that the info that we provided was helpful. Good luck with crafting your proposals on this call or in future ones. We look forward to reading them. Thanks a lot. Bye.